cloning, right, is eventually getting the actual full sequence. So again, when you're finding that cloning paper, make sure that you're seeing some screening of the cDNA library, and then I'm asking for the size of the cDNA, you know, the number of amino acids. So of course you need the sequence in order to know that information. If you're stuck on finding a mapping paper or finding a cloning paper, let me know. If you want confirmation that you're on the right track, I can do that before you upload it. I'll do it afterwards as well. It's okay. And if you don't have the right paper, it's not the end of the world for the draft, but obviously the sooner you find the right paper, the better for you. And then the last part is confirming that the gene they clone is correct. And for this one, you're looking for a paper where mutations are found in the gene in patients, or that they can rescue the normal phenotype in cell culture or in an animal model, or maybe they've done gene therapy, though that's less common because that's much harder to do. Or the third type of experiment would be like a phenocopy experiment, like we've talked about, where you use RNA interference and then you can see a similar phenotype. Or maybe you make a transgenic knockout mouse and you see similar symptoms as you see in humans. So those are the three types, and you need two of those kinds of experiments. Okay, so I gave you some hints in OMIM, like where to look for those kinds of papers. Again, if you're kind of getting stuck at all, let me know. And part 5 is the protein function in molecular pathology, and that goes along more with what we were talking about. For this, you may actually be able to use some review papers for the first part, like just like the normal function of the protein and how it changes after mutation. But for part D, you'll need another primary research article where I'm asking for one experiment where they investigated how either wild type or mutant protein functions in either a genetic model system or cell culture. Okay. So questions about parts 4 or 5? Anything been difficult as you've started trying to look for papers and work your way through? All right, I'm not hearing any questions. All right, hopefully it's going okay. I want to say one more thing about the deadline for this one. So if you find that you get through part 4, not quite through part 5 by Thursday, go ahead and upload part 4 and then finish part 5, you know, Friday or whenever you can get it done, Saturday. I'd rather see a complete part 4 on Thursday. And then, you know, a complete part 5, even if it's a day later, that's better, I think, than like if you just run out of time or you can't quite find those papers. So definitely focus on part 4 for Thursday, and I'll post this in Blackboard to let everybody know that you can have a little bit of wiggle room for part 5, but we don't want to wait too long, right? Does that make sense? Any questions about that? Okay, I'm not seeing questions, just a lot of thank yous. Okay, so I think, did I have anything else? I'm looking at my notes. I think that's oh, how do we find cloning papers if there's not a cloning section? Oh, okay. So importantly, remember that there's two, here we go. Let's go back here to the cloning, that there's two kinds of pages in OMIM. So there's the gene page, and then there's the disease page. So the gene pages have an asterisk before the, you know, number like 21375 or something like that. They have an asterisk. That's how you know you're on a gene page. The disease pages have a number symbol. At the very top of this, I just reminded you that there's an OMIM info PowerPoint where that's kind of explained. That's also in that research paper folder. So the disease pages, the ones with the numbers, are not going to have the cloning section, right? Because the disease isn't cloned itself, but it's the the gene page right here. So now I've highlighted, oh, you can't see because I'm not sharing. Oh, I am sharing my screen. Okay, sorry guys, I lost track. But can you see that? I've just highlighted, you want to make sure you're on the gene page. Okay, good. I know I said it 3 times. I'm just trying to be clear. Okay, if you still get stuck, let me know because I can, send you direct links, right? If you get stuck at any point. Other questions? Oh, what if the gene page doesn't have it either? Huh. I would, okay. You know what? Shoot me an email and I'll help. Is that Rebecca, right? Asking if you don't see that cloning section for the gene page, let me know and I'll work with you to make sure that we're finding the right paper. Okay. Yeah. Other questions about what we're working on? I'm going to tell you one more thing before we kind of finish off. Um, Google Scholar, yeah, right. That Miles is giving a tip that Google Scholar can be helpful, and so is PubMed. The only thing with those, I mean, well, they're great, so use them, and if you find the right kind of, you're able to find papers, especially if you don't have a lot of papers in your OMIM page, that's great. So yeah, reminder, there's multiple sources. The one thing I like about OMIM is that it has that table of contents, so that for most of you, you'll have a cloning section, and for most of you, you'll have a mapping section. So you'll know that you're getting to like the right category of papers. So I feel like that's pretty helpful from the OMIM standpoint. But yeah, if you're having any problems of any kind, just let me know. And especially if it's like a specific question, right? Like, it'll be probably easier just even to do it by email a little bit. Um, or we can have a private WebEx session, right? But we can, I can just kind of search around and help you find what you need. Um, Oh, yes. Okay, sorry. Lost my train of thought. So, coming attractions. So, the other thing I want to mention, obviously, this is what you're going to be thinking about for the next several days, is sections 4 and 5. I did want to just mention that after you work on sections 4 and 5, the last piece is the proposing your own experiments. And I just posted this assignment in Blackboard, and I know you're not ready to work on this yet, but I just wanted to let you know it's there. So this is the outline, right? This is part 6. So this, first of all, is an actual outline, so you don't have to have complete sentences. This is different from the first 2 drafts. This is where you're going to do your planning for for proposing these experiments. So you will find your table of techniques to be really helpful for this part of the paper. And I'm going to be posting a PowerPoint talking about gene therapy, CRISPR, and microarrays. Well, there'll probably be multiple PowerPoints there, like 2. And those are going to be really helpful methods where you're going to use all of these different techniques and methods that you've learned about throughout the semester, and you're going to propose some new experiments. And they need to address either diagnosis, how to improve it, treatment, again, how to improve it, or how to understand how the gene acts, how the protein acts, right? Either the wild type or the mutant protein. So undergrads, you're designing a total of 4 experiments, and they have to come from at least 2 of those categories. And then grad students are doing 5, and they need at least 1 from each of those 3 areas. And so again, this assignment's now posted in the assignment tab. And I've given you a structure for how to write these proposals. So you're just going to type right into this outline, right? So that it kind of has, like, so that it looks like an outline. So this, again, is different from those first 2 drafts. Any questions on anything I said so far? know you'll have more questions when you get to this part, which won't be till you finish parts 4 and 5. But anything come up right away as you consider what the assignment is? Okay. I'm not seeing a lot of questions. Any questions? All right, when you get to it, I think you will have questions, and that's fine. We'll just take them when you get there. But I just wanted to kind of let you know what's kind of coming. So I'll be putting up a narrated PowerPoint to explain gene therapy and CRISPR. I think that's going to be a single one. And then there'll be a separate one to explain what microarrays are and how you can use them. And then I'm also going to post some just like a Word document that kind of helps you think about how you would construct those kinds of experiments. That'll be really helpful for this section. Okay, so that will be coming attractions. We'll talk more about that later. Okay. So I think that was pretty much what I had planned for today, or what I wanted to talk about. Does anybody have anything else they want to ask about or comment on? Anybody else have inspiring words? Hard to top Queen Elizabeth, but you know, I think there's a lot of people trying. All right. No more inspiring words. All right. I hope you're finding ways to stay a little bit motivated. Hang in there. We only got 4 weeks to go. So we're almost there, home stretch. So most importantly, take care of yourselves. And again, feel free to one-on-one ask me questions for parts 4 and 5. All right. And have a good day, guys. Hey, Emily, it looks like it's just us.
Yeah, sorry, I'm pulling off the tape from there we go. So I am still waiting on a few of the articles that I need to write 4 and 5 to be given to me through interlibrary loans. Um, so I was wondering like um, what we, what I was supposed to do, because I can get a lot of it from OMIM, but the articles that OMIM used haven't come through yet.
Sure, sure. So when that kind of thing happens, just let me know. Like I was kind of saying, like I'm really happy to be flexible with these deadlines. I don't want you guys to get too far behind. So, you know,
Right,
I don't want to like say, oh, turn it in whenever, you know what I mean? Like because
right.
then we're never going to finish these papers. But if you don't have them yet, you can't write it. So that's
Right.
fine. So in that case, yeah, just let me know like you just did. And
Okay.
like, you know, if you like when you get them, like then once you get them, then let me know just so that I have an idea when to expect it. You know what I mean?
Okay.
So like
Yeah.
let's say
Yeah.
for example, let's say you get the papers late tomorrow, right? Obviously you're not gonna have it written by Thursday, which is fine.
Right.
So, you know, maybe by Monday or Tuesday,
Okay.
something like
Okay.
that. You know what I mean? But let's just stay in touch just so I know when to look for it.
Okay. And then when I do submit for 4 and 5, do you want me to include 1 through 3 then? Or because I'm rewriting it or
If you have it, great. Like if you have
okay.
those papers, sure. But if you don't, that's okay. Like
Okay.
no rush.
Okay. Okay. Yeah, I started to look the day that I read your email, I had started to look about it and I was like, well, I know what factor it affects, but they don't have anything else. And I was like, uh-oh, this is
Yeah.
not going to be good.
Yeah, I never actually heard of acquired hemophilia. So I
Right.
just, right. So I didn't realize like it's actually not like really
Right.
considered a genetic disease. And then as I was reading more, I'm like, oh, I see why. Like, it's not actually due to a mutation in the factor, you know.
Right. Right.
So
So
yeah,
yeah, I think, I mean, for the most part, it's pretty easy to find stuff on hemophilia A now that I've factored out the acquired part of it.
yeah.
Um, so I don't think it'll be too hard, but yeah, I'll let you know when I get those articles in.
Okay, sounds
Okay.
good. Yeah,
Thank
no
you so
problem.
much.
Oh, you're welcome.
Okay, I'll talk to you later.
Okay, have a good day.